I do want to talk about PARP and DNA repair, and more than just PARP with regard to DNA repair. We've heard it alluded to many times at this conference, and what I want to do is take a deep dive into considering why it is that we might target DNA repair factors in advanced disease. So something to consider is that every medical oncologist in this room actually has been targeting DNA repair, or at least androgen receptor directed mediated DNA repair, which we now know exists, which of course is targeted by the standard of care for patients with advanced disease. So as of late 2013, we came to recognize that the functions of the androgen receptor that we knew about in terms of promoting the expression of these secretory factors that are useful for us in diagnosing and managing and following management of prostate cancer, and also in inducing tumor cell proliferation and survival, is accompanied by a third mechanism, which is quite important, which is the ability of the receptor to induce pathways that promote DNA repair, in mostly double-strand DNA break repair, and resistance to DNA damage, and the knowledge that castration-based agents and AR-directed therapeutics antagonize these processes. So this was first reported in 2013. We demonstrated this. There's a companion brief from Charles Sawyer's lab that came to the same conclusion. Both groups became very interested in this pathway because we were concerned about a lack of understanding of how it is that hormone therapy acted in concert with radiotherapy for patients with locally advanced disease. And so both groups were chasing down that pathway and came upon this knowledge. In terms of the mechanism, which we chased down, we believe that the major mechanism by which AR promotes DNA repair is in fact through its ability to modulate the kinase DNA PK, which I want to talk about later in the talk. So targeting AR DNA repair crosstalk has become an area of considerable interest in the field. And I want to focus on two that are actionable, for which I think there are some significant underpinning rationales to target. The first is PARP1, which, as you may all know, modifies proteins by poly-ADP ribosylation. Something I want you to consider as we move forward is that PARP's most preferred target is, in fact, itself. So looking at PARP autoparylation is a mechanism for looking at PARP activity in tumor cells. The second factor I want to talk about is DNA PK, which is, of course, a kinase, well known for its role in controlling DNA break repair as part of DNA repair complexes, but which you may not know was actually first identified based on association with a transcription factor, SP1, and is well known to have roles on chromatin outside that of DNA repair. In fact, this is common to both PARP as well, PARP1 and 2, as well as DNA PK, of having these dual roles on chromatin as part of DNA repair complexes, as well as gene regulatory complexes. So PARP as a therapeutic target is something I think that we're all very interested in at this moment. And what I'll tell you, interesting Mac to PC conversion, but okay, let's all list these as number one in importance then. And what I'll tell you is I think there are rationale for three potentially different mechanisms. One is for PARP, of course, in combination with the genotoxic agent based on its role in DNA repair. In prostate cancer, there may be some particular usefulness as a single agent, especially in patients with DNA repair alterations, but also as a single agent or in combination for tumors that may be addicted to PARP pathways, especially those for which PARP is engaged in regulating the androgen receptor and DNA repair availability. So it's well known in other tumor types that PARP1 inhibitors work very well in tumors that have a BRCA1 or 2 alteration, or just an alteration that renders them defective in homologous recombination. And this is the classic synthetic lethality that we've all heard about. The concept is actually very simple. So in tumors that lack appropriate homologous recombination pathways, they have compromised double-strand DNA break repair, therefore making them more reliant on other pathways for repairing DNA as cells proliferate and divide. So PARP1 inhibitors antagonize remaining DNA repair pathways, and the concept is that you accumulate lethal breaks as a result of oncogenic stress and cellular proliferation that ultimately cause these tumor cells to die in the absence of a way to manage their complicated genomes in cells that are actively proliferating. And we now have proof evidence for this. We've heard at this meeting already reference to the TOPARP trial, which is still under embargo, so I can't show you the data from this trial, which is of course led by Johan de Bono. This is just an example from one patient on this trial, and I think we've probably all seen similar data where CTC count declines are remarkable in this patient with PARP inhibitors as a single agent, accompanied by this decline in PSA and radiographic evidence of response. This particular patient was really interesting in that the individual had to undergo a drug holiday for 5 weeks due to a non-therapy related issue. So really remarkable responses in this very difficult to treat patient population. Now, as we all know, some of the patients, a significant number of the patients in the TOPARP trial ended up having DNA repair alterations. And if we take a step back and we look at trials overall for multiple tumor types with regard to BRCA status and PARP, you see some interesting data. So in trials for which patients were selected based on a BRCA alteration or selected in ovarian cancer, for example, et cetera, you see that the responses are very good. However, it's the case, if we look at objective responses, that not all BRCA mutant tumors respond to a PARP inhibitor. So why is that? It's something that we can still debate. Further, if we look at unselected populations where all comers were accepted into trials, we see that, again, not every individual with a mutant tumor responded to the PARP inhibitor. And as well, there are a number of individuals who had wild-type BRCA status that responded to the PARP inhibitors as a single agent. So again, telling us that there are things that we really need to consider, biologies associated with PARP1 and PARP inhibitors that we may want to think about as we move these agents into the clinic. Further, this is another study from Johan de Bono's group that we were actually unassociated with, where there were a number of patients with advanced metastatic prostate cancer on the study. And in this particular study, he used a decline in circulating tumor cells as a surrogate marker for responsiveness. And as you can see, there are a number of individuals whose tumors were really indifferent to the PARP inhibitor, and others that were very good responders. And I should say these are patients who had had significant treatment with prior agents, including abiraterone, enzalutamide, and docetaxel. Now, the paradigm of who would respond didn't fit at the time. So these are all BRCA wild type. P10 status didn't matter, ERG status didn't matter. So things that we had identified preclinically as potentially a biomarker of response to a PARP inhibitor really didn't apply. Again, underscoring that we know some things, like if you're BRCA altered, you may be a good responder, maybe not. But certainly other factors may be playing into who responds to a PARP inhibitor and who doesn't. So what might be the basis of the response to response to PARP inhibitors in tumors that are proficient in DNA repair? And so this may become of particular importance in prostate cancer, especially for advanced disease, as it appears that a number of tumors have at least a hyperactive PARP pathway. So this is unpublished data from Matt Sheaver, who's a fellow who's here with a poster and will be presenting on the last day of the meeting. I don't want to steal his thunder too much, but by immunohistochemistry and looking at PARP activity through looking at autoparylation, what we observed is that a large number of these metastatic castrate-resistant tumors showed high levels of PARP activity compared to hormone therapy-naive tumors, and that this is associated with a poor outcome when using progression-free survival as an endpoint. So suggestive, at least, this is a relatively large cohort, needs to be validated in a second cohort, but suggestive that PARP1 activity is increased as a function of disease progression and is associated with poor outcome. So what could this mean? So a number of us got together, myself, Johan de Bono, Mark Rubin, and Felix Spang, some close collaborators, earlier this year rather, and put together everything that we could find in the literature about PARP1. What does it do? And how does this help us understand what's happening in patients who are experiencing or treated with a PARP inhibitor? So with regard to DNA repair, the pre-existing paradigms of what it is that PARP1 does in DNA repair was initially thought to be almost exclusively associated with base excision repair. We actually know that that's not true now that PARP1 has been shown to be involved in both single and double-strand DNA break repair through a number of pathways. And we wrote this review article on Molecular Cell that covers that. We also know through a number of other laboratory investigators that PARP1 has a significant role as a transcriptional modulator, including transcription factors like p53 that serve as tumor suppressors and pro-tumorigenic inflammatory transcription factors like NF-kappa B. And the transcriptional functions of PARP1 have been reviewed as associated with roles in cancer. And you can see from this list here, a number of transcription factors of importance for prostate cancer are known to be sensitive to PARP1 function. And with regard to the hormone-dependent cancers, the androgen receptor is in fact an important one of these. So it's well established that PARP1 has these dual roles, and could these be playing in? So there's some suggestions that this may be the case. So using patient-derived explants, which a number of laboratories are using now, including ours, where a tissue slice is obtained at the time of radical prostatectomy from patients who present with high-volume disease. The slice is subdivided and immediately cultured on wound healing sponges and then randomized to receive intervention or not across the protocol. And so in this case, the individual's tumor with high-volume disease that was treated in a control arm, or a subsequent slice that was treated with PARP inhibitor ABT888. We can validate that it's hitting its biological target through this reduction in PARylation. There's a tumor response, and that the slices that were randomized to receive the PARP inhibitor showed a reduction in Ki67. And then at biochemical analysis, a reduction in AR-dependent target gene expression. So validating again this concept that the PARP inhibitors as single agents are antagonizing the androgen receptor. So now taking then a step back and looking at what it is that PARP1 does as a single agent, looking at gene networks or transcriptome profiling immediately after exposure to a PARP1 inhibitor, we started to see a number of interesting paradigms. And again, these are unpublished data, and I would encourage you to go see Matt's poster and his talk to learn a little more. But what struck us in looking at this, and you can see based on the red stars, is that gene networks that control expression of DNA repair factors appear to be exquisitely sensitive to a PARP inhibitor. This is something we did not expect, and as you'll see when you go see Matt's data, it's the case that PARP1 inhibitors do seem to suppress the availability of DNA repair factors, most importantly, those associated with homologous recombination. So using unbiased analyses to ask, well, when we use the PARP inhibitor, what is the most critical gene network that's altered? It's the BRCA2 network. If we compare the gene signature that you obtain when you use a PARP inhibitor with a known deficiency signature for homologous recombination, these are almost overlapping, including those genes that are down when you use a PARP inhibitor, as well as those that are upregulated when you use a PARP inhibitor, indicating that PARP1 regulates availability of factors that are important for homologous recombination, again, in the absence of damage, and in the case of a PARP inhibitor as a single agent. So this leads to a number of potential predictions. So given the upregulation in PARP activity that we saw that was particular to metastatic castrate-resistant disease and correlated with poor outcome, are there PARP1 addicted subtypes with high PARP for which they're dependent on PARP1 for pro-tumorigenic signaling? And this could really manifest a pro-tumorigenic function in at least two ways. One, increased availability and activity of PARP and DNA repair complexes, allowing these cells to manage their complicated rearranged genomes that we know occur in prostate cancer. The other is PARP1 availability in transcriptional complexes that we know promote tumor progression, including the androgen receptor, but also potentially availability of homologous recombination factors, including BRCA1 and BRCA2, that can support the expression, support the survival of that tumor type. Now, the flip side converse is that a PARP1 inhibitor would be expected to induce an ineffective repair state, especially with regard to the factors, the repair pathways sensitive to PARP1, but also to weaken AR signaling, including the ability of AR to promote proliferation and repair, as well as to reduce availability of BRCA2 factors, combining to lead to damage accumulation and lethality. So what are the next steps for really testing this and what's happening right now in the clinic? So as I think we would all expect based on the TOPARP data, I think we'll start to see PARP1 inhibitors utilized for patients with metastatic disease who have known DNA repair alterations. That's expected from other tumor types, and I think would be a sensible approach. The second is an obvious need. Based on the data we looked at, I think it's important to identify who responds to a PARP inhibitor outside the obvious BRCA-altered patient, and this would include investigation of some of the pathways we just discussed. What is the level of PARP activity? What is the level of androgen receptor signaling? And what about the PARP-sensitive transcriptome that was identified? And lastly, rational combinations. So we know there are a number of trials ongoing at the moment. So Maha Hussein's trial with abiraterone plus or minus the PARP inhibitor ABT888 is still ongoing and is past the midpoint. PARP inhibitor plus radiation for locally advanced disease is planned by Felix Feng, and hopefully that will get off the ground. And then the trial we heard about earlier today, so I put in the trial schema, combining radium with PARP, which is PI of Kevin Kelly at my own institution in collaboration with those listed here. These are for patients who have seen either ABI or ENZA, not both, who have known bone disease, and it's a safety trial wherein some of the secondary endpoints will be to look at PSA declines, progression-free survival, and some of these same markers that we just discussed through biopsy, as well as through circulating tumor cell analysis. So with regard to PARP1, I think the path forward is pretty clear. But in closing, I want to put forward the concept of DNA PK as another DNA repair factor that we might consider targeting. Just to remind you of what I said at the beginning, DNA PK and PARP1 share some common features in that they both have very, very well-known, very well-established roles as transcriptional modulators on chromatin, as well as DNA repair factors on chromatin. And so it had been recently reported by our group and now confirmed through Felix Feng's group, and he's having a publication that should be submitted here shortly, that DNA PK, looking at an unbiased approach of what the function is of DNA PK is in a tumor cell, has a major function as a transcriptional modulator to drive pathways associated with tumor cell metastasis. And I won't go into what those are since the data have been published, but I do want to show you what the data are that we think support the next phase of clinical intervention. So in this case, we've taken prostate cancer cells and grafted them into, or given a tail vein injection. These are cells that spontaneously metastasize. This is the control arm of animals that received really no therapy. These are intact animals. That we can see the metastases develop after tail vein injection, unless these animals were treated with the DNA PK inhibitor, in which case you can see these tumors fail to metastasize, and this is the overall tumor burden. So in these data indicated to us, as well as others that are in the study, that DNA PK inhibitors suppress metastasis. Now, in profiling human clinical samples, we were really struck by the following piece of data. This is from a cohort, it's one of 3 that we profiled. This one's 545 patients diagnosed with high-risk disease and followed for 16.9 years. A third of them developed metastases over the time frame of being followed. And they were stratified based on their transcription profiling. And we asked GenomeDx, that had the cohort at the time, to help us look at all kinases. And what we saw in this red line right here is DNA PK. It's a top deregulated kinase in tumors that metastasize. We also went through with another cohort with our collaborator, Justin Drake and Owen Whitty at UCLA, and looked at DNA PK phosphorylation, comparing, and therefore activation state, comparing hormone therapy naive to metastatic castrate-resistant tumors, and saw a 40-fold activation of DNA PK in tumors that metastasize versus those that do not. So we really, we believe these data nominate DNA PK as a potential biomarker to predict who may metastasize or what tumors may metastasize. And if we look at outcome data from multiple cohorts, DNA PK or high DNA PK levels are strongly associated with shorter time to recurrence, shorter time to development of metastases, and poor overall survival. So these data indicate to us that DNA PK may in fact be an important target. We're moving forward with that. There's one agent available at the moment for clinical testing to target DNA PK. It's not yet been published. It's owned by Celgene, the compound CC115. This is a trial that we developed in collaboration with Dana Rathkoff, who will be the PI, Felix Feng, and myself, which will be run through the Prostate Cancer Clinical Trials Consortium, combining this agent with enzalutamide. Again, primary endpoint safety with secondary endpoints looking at PSA decline and anti-tumor activity. So key points here are really that I think understanding the basis of DNA repair factor function is going to be important for us to develop successful strategies to intervene with these factors in advanced disease. It's important to consider the full function of these factors as we move them forward into the clinic with dual roles in DNA break resolution as well as transcriptional regulation. It's notable that PARP1 activity as well as DNA PK levels are robustly enhanced in metastatic CRPC and appear to promote disease progression. Each again have these direct roles in DNA repair complexes, PARP1 has a secondary role in controlling AR-mediated DNA repair and BRCA1/2 levels. DNA PK, very differently, has a secondary function in promoting cell migration and metastasis through its transcriptional regulatory functions, and we believe these are targetable. And finally, identification of biomarkers to determine responsiveness to these agents, especially as single agents, is in progress. And hopefully by the time this meeting happens again, we'll be able to say something more meaningful about that. So I think I've thanked my collaborators along the way. I hope I got us a little more back on time, and I'm happy to take questions. Thanks. Uh-oh, here comes trouble.
Yeah. So just because I'm easily confused. So in the highly castration-resistant state,
Yeah.
is there cross-resistance to platinum agents and PARP inhibitors?
Yeah, I don't think we know enough about that yet. I think you're asking a great question. I don't think we've done the right studies to ask that question. With regard to any kind of preclinical modeling, even in prostate, I'm unaware of data that establishes that. So we have developed PARP inhibitor resistant models. We haven't tested them for platinum.
So, I mean, because I looked at some of the data on ovarian and breast, and every time I look at it, I get confused, and then people draw these various things about how DNA repair, and I can never get past the second step. But it seems to me that there is a certain degree of cross-resistance, and so I don't know where we go with the next step. Is the next step, you know, combining PARP inhibitors with other DNA damaging agents like cisplatin? Do we don't do them back to back? I'm trying to I just don't know where to go here.
Yeah, I think these are going to be important questions for us to understand, but to do that, I think we have to understand resistance to PARP inhibitors. So, and I don't think we're really at that stage quite yet.
And what about understanding resistance to platinum inhibitors, which, have been around for a while and sometimes haven't gotten as much, publicity because they're generic and nobody wants to talk about cisplatin anymore. But, there's some interesting data with those agents as well in advanced, disease.
So I totally agree with that, and I'm not afraid to use the C word. I'll say cisplatin. But the bottom line is, if you're the patient, which would you rather have, the PARP inhibitor or cisplatin?
I just want the one that works.
Right, exactly. I want the one that works too. But if you're, when you compare the tolerability and the side effect profile, I think the PARP inhibitor is a lot. I mean, from my perspective, especially as a single agent, is a lot more tolerable than cisplatin. So I don't have a prostate, so I'm going to say that as a disclaimer. But if I did, I think I'd rather have a PARP inhibitor than cisplatin.